before we get into the itty-gritty details of transformation, let me give you a quick overview. Transformation occurs when genes are transferred from one bacterium to another as naked DNA. It is when cells die and break apart, causing DNA to be released into the surrounding environment. Bacteria in close proximity to this free-floating naked DNA takes that DNA in and incorporates it into its own. This DNA that was incorporated has potential of having advantageous genes, such as antibiotic resistance. Now that you have a basic idea of what transformation is, let's dive into the details to show exactly how transformation happens. However, in order to know the process, you need to know the key players. We'll show you an image of the parts of the bacteria cell and beyond that are part of the transformation process. This way, when we talk about the process, you will understand which parts we're talking about and what we use their name. With the knowledge of what the parts are, we can now delve in the process. It all starts with one bacterial cell. Transformation cannot occur until this bacterial cell dies, which causes the naked DNA to be released into the environment, which is the bloodstream. In this case, the lack of oxygen in the bacterial cell causes its state of competence, leading to cell death. This now dying cell starts to break down, starting with its cell membranes, eventually leading to its nucleus. What is left is naked DNA free-floating in the bloodstream. This DNA continues to freely float in the bloodstream until a different bacteria cell comes upon it. Here, this other receiving bacteria cell is meandering through the bloodstream when it comes into contact with the free-floating naked DNA. From here, the bacterial cell goes to take in this DNA, which will eventually be incorporated into its DNA. But first, the DNA needs to get past the bacterial cell's membrane. The process for getting the naked DNA inside this other bacterial cell is called DNA uptake. So, let's take a closer look. We are now looking at the bacterial cell at a microscopic level. The free-floating naked DNA, when it comes into contact with the cell membrane, comes into contact with porous secretin. This prompts the pillins to come towards the cell membrane in order to connect in a way that acts as a tunnel for the naked DNA to enter the cell membrane. With the presence of ATP, which gives energy for the T4P motor complex to function, the T4P motor complex directs and builds the pillins across the membrane, reaching the outer part of the cell membrane close to the naked DNA. The DNA then attaches to the pillins on one side, which straightens out the DNA, slowly entering the cell membrane as the T4P motor complex breaks apart the pillins one by one. The pillins disperse into the cell membrane as they are removed, and as they are removed, it draws the DNA closer and closer since it is attached to the previous pillin tunnel. Once the DNA is within the membrane, another pillin tunnel is built with the T4 motor complex to the presence of ATP in order to push the DNA through the cell membrane and through the Rec2. The DNA then makes its way over to the Rec2, which is situated on the inner area of the cell membrane. As the DNA travels through the Rec2, parts of its other strand break apart and disperse into the cell. Now, only one strand of the naked DNA remains. The DNA, which is now in the cytoplasm of the cell, travels up to the nucleus of the cell, where the receiving bacterial cell's DNA is found. Recombination enzymes of the receiving bacterial cell will allow the now single-stranded naked DNA to bind to the receiving bacterial cell's DNA. The homologous to the naked DNA part of the receiving cell's DNA will also be removed so as to make room without adding extra DNA. Now, the DNA of the receiving bacterial cell has part of the DNA of the other bacterial cell. Transformation has occurred. If the DNA that was taken into the bacterial cell has antibiotic resistance, then it will also gain antibiotic resistance, allowing it to become stronger and eventually a superbug. Hopefully, you now understand transformation much better. Thanks for watching!